Thank you all for joining from various different time zones. We really appreciate it. So without further ado, I would like to introduce our first speaker, Dr. Jolanta Vidugrene. She is a senior research scientist at Promega in the assay design division of R&D, where she leads the development of a broad range of novel technologies. Her extensive expertise is in cell metabolism, cell viability, and cytotoxicity research. Dr. Vidugrene received her PhD from the Biochemistry Department, Vilnius University, Lithuania. She has a postdoc from Boston Medical Research Institute and also from the Rockefeller University.
Okay, so hello and thank you for joining us. It is my pleasure today to present our work on developing bioluminescence approaches for studying a very dynamic and very interesting area of cellular energy metabolism. So as we all know, healthy cells in culture constantly consume nutrients, use them in different metabolic pathways, and secrete byproducts back into the media. So it is an important function of the cell since it allows cells to grow, maintain function, and respond to internal and external signals. But what it is important that different cells have different function. Therefore, metabolic pathways have to adjust. For example, if we look at cardiomyocytes, they have a high demand for ATP and therefore rely on mitochondria function. For adipocytes, the main function is to store energy. Therefore, they rely on completely different metabolic pathways such as lipogenesis and lipolysis. Naive T cells show very low nutrient uptake, but once they activate it, they have to change the entire metabolism to support a rapid proliferation and growth. Also, cells don't work in a vacuum. They always have to interact with each other, and therefore metabolic pathways have to adjust depending on the changing microenvironment. Also, they have to work in sync with epigenetic and signaling pathways. And misalignment between those three pathways often leads to metabolic disease or different disease states. So that's why we are interested in studying cellular energy metabolism and getting more information about connection between metabolic state of the cells and different epigenetic signaling pathways. So how can we study cellular energy metabolism? So if we look here, multiple approaches are available. So including metabolic profiling using analytical methods, stable isotope labeling for studying metabolic fluxes, or measuring oxygen consumption and media acidification using, for example, CHOS technology. All of those approaches are informative, but require special instrumentation and not always readily applicable to 3D cultures. So one of the approach that does not require special instrumentation and is based on individual metabolite detection using coupled enzyme reactions. So therefore, when we thought about building our metabolism portfolio, we took exactly this approach and built our assays using coupled enzyme reactions. But we also took advantage of sensitivity and dynamic range of bioluminescence readout. So what we use in our assay development is a novel proluciferin that is a reductase substrate that is in the presence of reduced dinucleotides is converted to luciferin. And then luciferin is used by luciferase to produce light. So now by using this core technology and coupling it with enzymatic metabolite conversion, for example, converting glycogen to glucose or glutamine to glutamate, and then adding another step, we're using a metabolite selective dehydrogenase, we can build a set of metabolite detection assays that are shown here. And the list is continuing to grow as we continue to develop. So one of the advantages of those assays that they provide a really quantitative metabolite detection and easy to use. So detection reagents specific for each metabolite are made, added to the sample at 1:1 ratio, and luminescence is red. And as shown here, they can rapidly apply to different assay formats, for example, 96, 384, 1536 well plates. They also can be used with different samples like tissues, cell cultures, blood, yeast, bacteria. So that's provide a very wide application of those assays. So what information we can get of using those metabolite assays? So for example, here is one example that it offers a convenient tools for measuring metabolic phenotype of different cells. So it is well known that rapidly growing cells increase glucose consumption and lactate secretion. They also depend on glutamine metabolism, and the activity of both pathways can be measured using metabolite assays. So the assays also are well suited for inhibitor screening in a high throughput format, as we have described in this paper. But one of the advantages of this wide applicability of those assays that they also can be applied to complex biological models, as we will hear from Katie in the next talk. And they, for example, can be used for studying insulin action in diabetes or measuring accumulation of triglycerides in NAFLD and NASH models. So next, I would like to go into more practical details about how do we use those assays, as well as a general consideration for setting up metabolism studies. So as shown in this slide, metabolite levels in different samples can range from millimolar to nanomolar range. So therefore, taking into account the linearity of metabolite detection assays is very important. So if you want to study changes in metabolites in the media, so it is a very simple protocol. So you just collect the media at a different time points. The media can be frozen since metabolites are stable. And then after proper dilution to fit the linear range of the assays, samples can be split and multiple metabolites can be measured, as I will show in the next slides. But also, the sensitivity of bioluminescent assays allows to measure intracellular metabolite levels. And that can be done since our detection reagent contains a very high level of detergents. So by adding the detection reagent directly to the cells, the cells are lysed, the metabolism stopped, and we can detect the intracellular metabolite levels. But another very important consideration for studying cellular metabolism is, as shown here, media composition. As we all know, different media contain varying amounts of nutrients that directly affects cellular metabolism. For example, glucose concentration can range from 5 to 25 millimolar, 5 millimolar being more physiological, since normal glucose levels in blood range from 3.5 to 5.5 millimolar. So therefore, by setting the metabolism study, keeping the media as close to the physiological state is important. We also suggest using, if possible, media that doesn't contain nutrients, and that gives a flexibility to add a required nutrients at a concentration you like. But also important to consider that serum that we often using in our cultures contains a significant amount of different metabolites and other small molecules. So therefore, using dialyzed serum can give a more controlled situation when studying metabolism. But another thing to consider, as I show in the next slides, that not only the media affects the metabolism by the cells, but it also affects the sensitivity of detecting changes in metabolite levels. For example, as we show here, we compared glucose consumption and lactate secretion during spheroid formation in two different media, in a low glucose 5 millimolar and high glucose 25 millimolar. So here HCT16 cells were plated at different cell densities, and after spheroid formation, we collected the media and analyzed. And as you can see, in low glucose media, the glucose level decreased with increasing cell numbers and could be measured with as low as 1,000 cells per well. In high glucose media, as we could see a cell-dependent decrease in signal. However, statistically significant decrease only could be measured with the higher cell numbers, let's say 5,000 and 10,000 cells per well. So this data show in a relative light units, but now if we can take the light units and convert it to the amount of glucose that those cells consume, we can see that the cells growing in high glucose media showed about twofold higher glucose consumption. But as we can see that even at 5,000 cells per well, it only corresponded to 17% consumption as compared to the starting glucose concentration. And if you have a lower cell numbers, the amount of glucose consumed was below 10,000, and that is with a standard deviation of the detection. So therefore, using lower glucose, we could see a much more reliable changes and could measure it with a lower cell numbers. So next, we used the same samples and analyzed the secretion of lactate. So in general, media doesn't contain lactate. However, significant amount of lactate is present in serum, and it is dramatically reduced, as I already mentioned, in dialyzed serum. So therefore, for example, if you are setting up an experiment to look at the lactate secretion and have 1,000 cells in 100 microliters, so you can see about twofold signal increase above background in 1 hour using dialyzed serum, when it will take 100 hours for the cells to secrete enough lactate above the background to see the difference. Also, setting at lower volumes of the media improves further the sensitivity. So it also the same is true as it's shown here when we looked at the lactate secretion in the formed spheroids. Again, by using dialyzed serum, we could see a nice increase in lactate as compared, to the non-dialyzed serum. And the data here are shown for lactate, but it is true for any metabolites that are present in serum, for example, glutamate or glutamine. So using a dialyzed serum, helps a lot when you are studying metabolism. So next, of course, we wanted to evaluate if metabolic activity of spheroid changes with size and can it be inhibited by a known metabolic pathway inhibitors. So in this case, we used a metabolite preferred media that I described as 5 millimolar glucose and also, dialyzed serum. And as shown here, this media supported the formation of spheroids, and the different sizes of spheroids were formed depending from the number of cells that were seeded. So now if we look at the metabolism of those cells, as we see here that as the cells continue to grow, even after formation of spheroids, they have a relatively high consumption of glucose. They also secreted a lactate at a very high rate. And when we calculate, about 80% of consumed glucose was converted to lactate, suggesting glycolytic phenotype of HCT11 spheroids, which is expected from a cancer cells. So now, we also took the same samples, the same media, and looked at the glutamine metabolism. So the glutamine metabolism was less clear. As is shown here, spheroids growing at slower rate show a slight decrease in glutamine consumption and increase in glutamate secretion. However, more systematic analysis is needed. But what it was clear that, as I showed previously, about 80% of glucose was converted to lactate and lactate was secreted. In this case, only 4 to 8% of the amount of consumed glutamine was secreted, was converted to glutamate and secreted. So that suggests that the cells use glutamine maybe in a different pathways and retain it inside the cells. So now, can we use the same approaches to screen for the inhibitors? That is important in determining the activity of metabolic pathway. So to test if glucose and glutamine metabolism can be inhibited in 3D cultures by a known glycolysis and glutaminolysis inhibitors, we treated spheroid with 2-deoxyglucose, a non-hydrolyzable glucose analog, a known glycolysis inhibitor, and we also used BIPTEST, a selective glutaminase inhibitor that inhibits glutaminase conversion to glutamate. So the media samples were collected, and after 24 hours of treatment, and the change in lactate and glutamine concentration was determined. Also, in this case, we included a real-time glow viability assay to look at the changes in viability. Real-time glow viability assay is non-destructive, non-lytic assay to measure the changes in viability. Therefore, it's very well fitted, to be included in a different metabolism studies. So, and as shown here, under the used experimental condition, no drug-induced toxicity was measured, but about 90% of glucose-dependent lactate secretion was inhibited by 2DG, and 65 to 70% of glutamate secretion was inhibited by BIPTEST, a no glutaminase inhibitor. So taken all together, this data indicate that luminescence metabolized assays can be applied not only for measuring metabolic changes in 3D models, but also can be successfully used for metabolic pathway inhibitor characterization and screening. So in the last few slides, I also would like to make a few comments about using the assays for measuring intracellular metabolites. So this graph illustrates the range in metabolite concentration in cancer cells. And as you can see that intracellular metabolite concentrations can change from millimolar to nanomolar range. But since our assays measure intracellular metabolite concentrations after lysing, we have to consider that once you lyse the cell, due to the increase in the volume, the metabolite concentrations in the lysates are in a low micromolar range. So therefore, you need a really sensitive assay to detect the changes in a small amount of cells, which fits well with the sensitivity and dynamic range of bioluminescence detection. So therefore, in using our system, we can measure intracellular levels of metabolites directly in 96 or even 384 well plates by just adding the detection reagent to the cells that lyses the cells and detects the changes. So here is just an example of using triglyceride glow assay for measuring changes in intracellular triglyceride levels in liver microtissues. The data were generated in collaboration with Inspero and show that accumulation of triglyceride levels can be increased by different treatment, for example, in the presence of fatty acids or LDL particle. So in summary, what I showed today that bioluminescence metabolite assays provide rapid and easy to use quantitative, that is an important, and quantitative detection of changes in metabolite levels and can be used both in media or inside the cells. Also, cellular metabolism can be studied using individual spheroids. We can study the changes in metabolites using as low as a spheroids from 1,000 cells. And also the sensitivity and detection can be increased by static experiments in 384 well plates, or as I showed before, decreasing the media volumes. But one thing that I would like you to remember from my presentation today is to pay more attention to the media composition. It is not only important for the metabolic changes that you are studying, but also it is important for the detection of the changes in metabolites. So decreasing the starting amounts of metabolites in media will increase the sensitivity of detection. And it can be done, let's say, by, as I showed, by using the media that contains low amount of glucose, or also using a dialyzed serum that doesn't contain a significant amount of metabolites. But one of the challenges associated, when you are working with a 3D cultures, that sometimes it is very difficult to remove the complete media. And if you think, for example, the cells are continue to secrete metabolites for 24 hours or 48 hours, and then you are trying to remove the media, but some media is left behind, that already will contain a significant amount of metabolites and has to be taken into account when you're calculating the changes, but it also most likely will reduce the sensitivity of detection. Therefore, considering different systems that will allow the complete media removal can be beneficial when looking at metabolism, changes in metabolism. So, and in the end, I would like to acknowledge the members in my group who did all the work. I also want to thank collaborators, early testers, and thank you for listening. And I think I'm ready to answer the questions. Thank you.
Thank you so much, Yolanta. That's a wonderful talk and really very clear. And appreciate the comment about being careful about the media composition, and especially when you're trying to measure metabolism. And I would like to introduce our next speaker, Dr. Katya Nikolov. She's a physician scientist currently leading the applications team here at Protein Fluidics. Her expertise is in 2D and 3D cell-based assays for phenotypic screening in the fields of oncology, tumor microenvironment, immunotherapy, projects such as T-cell-based assays and drug screens with solid tumor models. She has extensive experience in high-content imaging. She received her MD degree from the Russian National Research Medical University and pursued her postdoc work at Memorial Sloan Kettering.
Okay, so thank you, Yolanta, for the great talk, and thank you, Rashmi, for an introduction. So in the second part of today's webinar, I will talk about the platform that we have developed for 3D, for the automation of 3D assays. And I will share with you data that how we approach multifunctional profiling and metabolite analysis using patient-derived tumoroid using our platform. So first, I'd like to acknowledge our collaborators that we work together with. We work closely with Yolanta Group at Promega on optimizing the luminescence-based assays that she talked about today for the use in 3D models in our Tumor system. We also work closely with Dr. Burrow's lab at Tulane University, and their research focused on developing triple-negative breast cancer patient-derived models. And we work together using these models to look at the drug response profiling and optimizing and developing assays, again, using these models for the use in Tumor system. We also worked with Greiner Bio, and we like their technology, nanoshuttle technology for magnetization of the cells, and we adopted this technology in our platform, which I will be talking later in more details, to facilitate the positioning of the structures inside our plate. And we work also closely with Molecular Devices Group. They help us with confocal image acquisition and data analysis. So today, first I'll briefly talk about why we think moving to 3D cell models is important. Then I will do an overview of our platform, Tumor system and Flowchips, and how it works. And then I will share with you our work that we have done on optimizing luminescence-based assays for viability and for metabolites analysis in 3D models. So before we go into assays and data in details, I wanted to briefly highlight why we think moving and doing these experiments in 3D cell models is important. So we talk these days more and more about precision medicine, and interestingly, the term personalized or precision medicine has pretty much become a synonym of genomic analysis, which is used to identify the right drug for the right patient. Now, the challenge is that only a small fraction of patients actually carry actionable mutations and benefit from such approach for the selection of the appropriate treatment for them. And we think that actually cancer is a much more complex system than that. And what we really need is to embrace functional and phenotypic profiling, including genomic analysis, based physiological 3D models that recapitulate the whole complexity of tumor microenvironment. So why this is important? Because tumors are not only genes or not only comprised of the layer of the same cells. Tumors have phenotypically different cell populations within the extracellular matrix, which is also functional and active. And all these cells and all this part of the tumor contribute to microenvironment and even show differential responses to therapy. And so to solve the cancer problem, we really have to address this as a whole system. Now, I mentioned that we collaborate with Dr. Burrow group at Tulane University, and they use highly translational patient-derived xenograft models, which really has transformed the field of breast cancer research and drug discovery. The workflow they have developed and established in their lab starts with acquiring patient tissue biopsies, which is then processed, and the extracted material they use to propagate in mice, and then characterize these models using genomic sequencing and histological approaches. Now, lately they try to move to more scalable tumoroid models, which we call it, and in our work we use primary cells that they developed and create tumoroid models out of these cells. And the data I will be talking about today will mostly focus on using these models. So now in the next few slides, I will give you a brief overview of our technology and how it works. So here you can see how the device that we design actually looks like, and you can see the relative size of it. Puma platform is a microfluidic platform that we designed to automate complex multi-step 3D assays, and you can run your experiments in medium throughput scale and importantly in physiological environment. This platform can be used as a benchtop, or you can put it inside the incubator, and in this case all the assays that you run will be performed under standard cell culture conditions. The heart of the system is the flow chips. Here you can see the holder with 4 flow chips placed in the holder. You can use between 1 and 4 depending on the configuration of your experiment, and we can fit 32 samples in these 4 flow chips. The dimension of the holder and dimension of the flow chips and distances between the wells resemble standard 384 well plate, which makes it absolutely compatible with instrumentation like plate readers or microscopes or high content imagers. So if we look at the design of the flow chip, we have 8 test lines in each flow chip, and each lane is designated to 1 sample. You can see the central well where actually we load the sample. It could be organoid, spheroid, or tumoroid. And then this well is connected to a number of reservoir wells through the microfluidic channels. If we look at the sample well, the size of it is relatively small. The volume is 20 microliters. And you can see that the sample is actually positioned inside the protected sample chamber. And the bottom diameter of this protected sample chamber is 1.2 millimeters. It is optically clear and suitable for high-resolution imaging. And so essentially what we can do is medium exchange in and out through these channels that you can see on this picture without disturbing the organoid or sample that you're working with. Now here I wanted just to show you a brief video. Here you can see how we load the sample inside the protected chamber, which is our proprietary technology. And we can perform media exchange in and out, keeping our sample undisturbed. We can do up to 95% of media exchange. However, if for any application you need to drain this well completely and remove media like up to 0%, you can also do so using the bottom of the through the bottom of the flow chip. So here I mapped out the workflow that we have developed for the Puma system. The first step would be, like I said, to load your material and all the reagents inside the flow chip. And you can load your reservoirs with medium, with different solutions with drugs. It could be staining solutions or lysis buffers, as long as the properties of this fluid is compatible with the microfluidic channels. Then this plate is loaded into the Puma system. And like I mentioned, many experiments that we do are being done inside the cell culture incubator, meaning that all the assays are being run under standard cell culture conditions in terms of temperature, humidity, and CO2 concentration. And then all you need to do is just select the preloaded protocol from preloaded list already on the machine. press run button, and then the assay will be running automatically. Once the protocol is completed, the plate is ready for the analysis. And for that, you can use you can do fluorescence microscopy, you can do luminescence readouts using plate reader. You also can do screenings using high content imager screeners. And if needed, you also can collect supernatants throughout the experiment and then either freeze them or process them for the downstream analysis for metabolites, for example, the workflow that I will be talking about today in more details. So here we demonstrated the utility of the Puma system in different research areas. We've done a lot of work with toxicity testing when we create spheroids and then expose them to drugs, different concentration of drugs, and then we stain them for viability, do data analysis, and translate this into dose response curves. We also have done work with a number of different organoids like intestinal or IPC-derived gastric organoids. We also used more complex models like microbrains, and we looked at, for example, calcium oscillations, the process which is important in neuronal firing. And today, the data I will share with you will be mostly focused on breast cancer patient-derived tumoroids. And then at the end of the talk, I will briefly talk about mass spec approach in melanoma. So how do we form tumoroids for our platform? Now, there are a number of different ways to do that. One of the approaches that we use is using agar-well plate. This plate comes in different sizes. We typically would use 24-well agar-well, which each well of the well plate contains 1,200 microwells, and each microwell has size of 400 microns. So to create tumoroids, typically we would create a cell suspension, and then we would seed the cell suspension to the well so that we have between 1,000 or 2,000 cells per microcavity. And then over the period of 24 to 48 hours, we see the formation of nice, tight, and pretty uniform tumoroids. So for the experiments, first we would retrieve these tumoroids from these microcavities. We also use nanoshuttle technology for certain applications to magnetize our tumoroids, and then we load them into the Flowchip using this magnetic load tray that we have designed. Here you can see how these tumoroids look like at the bottom of the Flowchip. You can use either one of them or you can load multiple of them. And magnetization approach helps us to pull these tumoroids down to the bottom of the protected sample chamber and to kind of more precise position them in the middle of the well, which makes it easier to image and advantageous for data analysis. Here, I would like to bring your attention to the concept of multifunctional profiling, where this platform provides substantial value because we can multiplex a number of complex and multi-step assays using individual sample and collect lots of data points. So here I show you schematically an example of the study that we've done using breast cancer patient-derived tumoroids, which we would load in the plate. And then we exposed them to a number of drugs at different doses. And then while the treatment is happening, while these tumoroids are exposed to drug, we collected supernatants for the downstream metabolite analysis at different time points. And then once the treatment is completed, we stained the cells for viability and did confocal imaging. And then because we're also interested not only on endpoints like viability endpoints, but we're also interested to look at different biomarkers, we did the immunofluorescence staining for these biomarkers to understand how expression level changes after exposure to the treatment. So today, I mostly will focus on these two parts, viability assessment and a metabolite analysis and the assay development that we've done for the Tumor system. When we do profiling of our tumoroids, for example, for the drug response, the first thing that we would do, like I said, we would first load the Flowchip with the sample and reagents. And then even before we expose this sample to any drug or do any interrogation with this, we first run real-time glow assay to assess pretreatment viability of the sample. And also it helps us to understand how uniform the tumoroids are. It can be used also for quality control of the loading. And these measurements can be used, for example, normalization of the cell number at the beginning of the experiment. And I will talk later about details of how we do these assays and how it works. Now, during the treatment, we collect supernatants. We can do it at multiple time points. And then at the end of the treatment, we retrieve the supernatants, and you either can freeze them until further use, or we perform, for example, lactate glow assay, which Jolanta talked about, to analyze the level of lactate secretion. At the end of the treatment, we typically would do endpoint viability assessment, again, by either staining and doing confocal measurements, or alternatively, we can use ATP-based luminescence cell titer glow assay and do measurements using plate reader. Again, I will be talking about this assay in the next slides. So, like I said, real-time glow is the first assay that we do before exposing our sample to treatment. The way this assay works, it is based on constant presence on viable cells. And then what we need to do is just add reagent to the cells inside the Flowchip. This reagent contains pro-substrate, which then is taken up by the cell. It is reduced to a substrate within the cell, and then this substrate reacts with luciferase and emits light. So essentially, if we have viable cells, which constantly metabolically active and produce light, we have a lot of luminescent signal to detect. And then if your cells are dead, they're not metabolically active anymore, and we don't have light, so you see the differences in luminescence signal. Doing these assays is relatively straightforward in 2D culture. However, once we move to 3D models, factors like sphere size or reagent penetration come into play. And so first we wanted to evaluate the performance of this assay in a 3D setting. And we looked at how luminescent signal would correlate with the density of the spheroids that we work with. And for this, we've done an experiment with HCT116 spheroids, which we created using different starting cell density. And here you can see that we have pretty nice linearity between seeding density and size of the spheroids and a luminescent signal that we detect coming from these spheroids. Then we wanted to look at how stable the signal would be as we culture spheroids. And for that, we created spheroids using 2000 MCF7 cells. What we observed is that it takes some time for reagents to penetrate inside. So we start to see specific signal, which is above the background at 2-hour time point of incubation. And then over time, the signal is pretty stable. However, notoriously, these tumoroids or spheroids are not exactly the same, so we see some variability between them. Now moving forward to tumoroids, well, it's kind of the same question. Well, how this assay will perform in our patient-derived tumoroids? And again, we saw pretty nice linearity between number of spheres versus luminescent single first and 384 well plate. And then we evaluated if we can measure real-time glow signal directly in our Flowchips. And like I said, our Flowchip can accommodate 32 samples. So we took these 32 tumoroids and then ran real-time glow assay on all of them. And first of all, we saw pretty nice signal-to-noise ratio, pretty high. And you can see that the data points between tumoroids are pretty close to each other. So we observed some variability, but overall, we were pretty happy to see how the data looks like. At the end of the treatment, one of the ways to do viability detection is to stain them for example, membrane integrity or use calcium AM staining. We have published these protocols optimized for the use in human system, which you can download today during the webinar. But an alternative way to do this is to use a cell titer glow viability assay. Now, this assay requires lysis of cells at the end. It's an endpoint assay, and it relies on constant ATP production by viable cell, and then on the enzymatic reaction with luciferase and substrate and producing light. So essentially, if you have nice culture of viable cells, we will see production of light. And once your cells starting to die, they're not anymore metabolically active, and they decrease the production of ATP. And of course, we see this in a decrease in light. So here I'd like to share with you an example of the data that an experiment that we've done with breast cancer patient-derived tumoroids. In this experiment, we treated them with 3 different drugs, romidaptan, trametinib, and paclitaxel. And then at the end, we measured viability using cell titer glow assay just by simply addition of this reagent directly to the Flowchip and to our sample, and we performed measurement of the luminescence in the Flowchip. And so here you can see a nice concentration-dependent decrease in bioluminescence, reflecting decrease in ATP production due to cell death. And also it's always nice to correlate with standard like confocal imaging when we use membrane integrity stains, and we also can see that we have increased dead color here, which indicates cell death, which correlates with cell titer glow. So the data shows you that it is possible to use this luminescence-based assays, real-time glow that I talked about before, and cell titer glow directly in the Flowchip without any need to remove samples and transfer it somewhere else. So now let's talk about metabolite analysis and how we approach it in a 3D setting using Puma system. So why this is important? It was almost 100 years ago when Otto Warburg described the phenomenon of the enhanced accelerated conversion of glucose to lactate in malignant tumors, even in the presence of abundant oxygen. And throughout these years, the interpretation of this phenomenon has been debatable. However, it is pre-accepted that for the majority of the tumors, aerobic glycolysis, even in the presence of oxygen and in the presence of functional mitochondria, actually constitutes a major driver of the cancer progression machinery, and also it was linked to resistance to conventional therapy and, unfortunately, poor patients' outcomes. Lactate is one of the very interesting metabolites that we're looking at. For a long time, lactate was only recognized as a metabolic waste product derived from aerobic glycolysis. However, it's not actually true. It has now been firmly demonstrated that lactate can be also incorporated into the TCA cycle. And now we know that lactate plays a role in tumor progression, it promotes invasion and metastasis, and it also was linked to drug resistance. Coming back to our metabolic assays, metabolic changes are usually quite dynamic, and therefore it is important not always to look at the endpoint, like when, for example, we kill all the cells with a drug, but to assess at earlier time points and look at actually the dynamics, how, for example, the secretions of the metabolites happening dynamically once we expose cells to drugs. So the beauty of our platform is that we can set the schedule for automatic sample collection. And here I wanted to show you a brief video so that you have better visual of how actually transfer is happening inside the Puma system. So if you look at this pink well that we have here, first we transfer medium to cells, incubate over time while cells keep secreting metabolites, and then we collect supernatant, transfer it back to the reservoir, store for until we completed the assay, and then we can perform the same transfer using another well. So essentially what's happening is that we constantly expose our cells to the drug, and at the same time we can collect number of supernatants using intervals that you would like to use in your experiment. So just fully customizable. So here just an example again how it can look like at the end. So once during the treatment, once all the supernatant are collected, we have them stored in all this reservoir as well at the end of the experiment. And then all you can do is just take them out and process for the downstream assays and do your readouts using plate reader. Now you can think of that maybe 20 microliters is kind of small volume. But actually, it's not true. 20 microliters is enough, and we have enough of secreted metabolites there to measure. And even quite often, we need to dilute it even more to be able to stay within the linear range of the assay. So it's pretty nice and sensitive assays, and you can do multiple of them, looking at the dynamic of multiple metabolites using only one supernatant sample. So here, talking about sampling, I'd like to talk a little bit more the method that we use in Puma system. So the standard way of sampling is to put your sample spheres or cells into the well where you have substantial volume of media, and then do small sample collections like 5 or 10 microliters, which will not significantly affect the whole volume of the well. And by doing so, we can look at the depletion of the glucose levels, for example, because they're being consumed by the cells. Or we can look at lactate secretion that are, again, constantly being secreted by the cells. Now, the interesting part of that is that it is this type of approach can be nicely used to look at the depletion of the components from the media. However, because the cells keep constantly producing these metabolites, when we look at the secretion components, we only have kind of small window of detection using this type of sampling. Now, in our Puma platform, because we do a full exchange of the medium and we actually collect almost the whole well as a supernatant, we do what we call discrete metabolite analysis. So the advantage of that is important, because, especially for secreted factors, because, for example, for lactate, we now increase substantially the signal that we detect above the minimum background. And so if we model signal to signal and background, how it can look like using these different approaches of sampling, with integrated sampling, it will give you a better measurement of the depleted factors. However, you have a limited sensitivity for the detection of secreted factors. Whereas using discrete sampling, it is much more beneficial for the detection of the secreted factors because we have substantially higher signal to background ratio. Coming back to our assays, we also optimize the lactate glow assay for the use in Puma system for our tumoroid experiments. And again, the first thing as part of the evaluation of this assay, we looked how luminescent signal will look like in our tumoroids in the Flowchip. And again, we see pretty nice linear correlation between number of spheres that we experiment with and luminescent signal coming from the Flowchip. And we have pretty good signal to noise ratio. So we're pretty happy to see these results. And then I'm showing you here the example how the data can look like. And these tumoroids were treated with 3 different drugs. And then we looked at lactate secretion dynamics from these tumoroids. In these plots, we showed the correlation between drug response and luminescent signal that was acquired at supernatant, acquired at different time points. And interestingly, because we did this assay within the first 24 hours of treatment, we didn't see substantial killing yet of the cells. So you can see on these images, we see a lot of viable cells. However, we observed the pattern of increased lactate production once we start exposing these tumoroids to drugs. We need to do more research to understand, biological implications of these observations. And also we know that lactate was linked to resistance mechanism, for example, in breast cancer. However, this data highlights that you can use this type of assays to understand how metabolism shifts during the exposure of your sample to drugs. And also it can be useful, for example, if you would like to screen drugs specifically affecting metabolite secretion or metabolic pathways, you can use them as well. And so in the last part of my talk, I would like to briefly tell you about how we approach sampling using Puma system if you would like to do mass spectrometry profiling of your sample. The approach is quite similar because we perform all our transfers automatically inside the device. We can load in a similar way our Flowchip with your material, and then we can do labeling and compound treatment automatically inside the Flowchip. We also can perform in situ lysis in the Flowchip, so you really don't need to take your sample out because any perturbation can affect metabolites, and it's very beneficial for obtaining accurate results. And so first we'd like to see what level of sensitivity we can achieve. And we've done this experiment when we took spheroids, which were created out of 2008 CT116 cells. We were able to profile these spheroids for 250 different metabolites. And here you can see, by using principal component analysis, nice clustering of your database on these profiles. These spheroids were either left untreated or were exposed to 2 different drugs, and you can see a nice clustering of data depending on the condition that we use. And then this set of data is coming from Sean Morrison's groups. They kind of use similar approach. They work with melanoma tumoroids, and they use multiple of them for that particular experiment, but you can use even one if you would like to. So in similar way, these samples were exposed to drugs, and then they looked at metabolites profile. And again, you can see nice clustering of data here between control group and treated group. To summarize, today I emphasized the importance of 3D models, why they think they're more physiologically relevant, and it's much better to use these models for your experiments and for disease modeling and drug responses. We see actually our system as open lab on a chip technology, which can incorporate different types of samples. It could be spheroids, organoids. It can be also used with extracellular matrix. Today, I showed you relatively simpler model, which is tumoroids derived from primary cells. However, we also work with a more complex model, and this work is under development using co-culture systems. And also we adopt these assays for the use with extracellular matrix. Of course, again, I would like to highlight the unique capability of our platform to do multifunctional profiling, which I think is quite valuable. And because we can do this automated fluid transfers, we can combine a number of different assays. Like I showed you today, we can do biomarkers assessment and metabolites dynamic assessment, and doing different functional assays together using individual sample and collect a large number of data points. And also, I think the assays that we talked about, luminescence-based assays are also quite valuable because they're relatively easy to use, and at the same time they're quite sensitive, and you can incorporate all of them in the workflow using Puma system for metabolites profiling of individual samples. I also would like to bring your attention that we have a certain publications and material available for download, where you can take a look in more details at the data we talked about today. Please reach out to us if you have any questions, or if you would like to discuss the development of your assay of interest using the platform. And I think I'll stop here, and I would be happy to take questions.
Thanks, Katya. There are a number of questions for both Katya and Jolanta. Let me start with Jolanta here. One of the questions you got from Evan is, can you customize your metabolite glow assays for other analytes?
Yes, we do. And, as I mentioned, we already have the list of probably 10 or 15, and the list is growing, and we can build on that platform. Since we have a core technology, we can continue to build.
Okay, cool. This is another one for Jolanta. Are the metabolite assays compatible with downstream RNA profiling or genome analysis?
Yes, they are, because most of the measurements are done from the media, so they can be compatible with any downstream application, viability measurements, RNA analysis, DNA analysis, imaging, as it was shown here.
Okay, and one more for you, Jolanta, before I pass on to actually, I have a couple of questions for both of you actually as well. So how do Promega metabolite assays compare to Seahorse analytics?
So Seahorse analytics is, it's always a difficult question, and it has a very good sort of way to measure oxidative phosphorylation, looking at oxygen consumption rates. And it is a snapshot of the current state of metabolism of the cells. So what the metabolite assays provide is probably more wider sense of what is the difference between different metabolites, since we can look not only of two pathways, which is oxidative phosphorylases and glycolysis, that is looked in Seahorse, and also we can see how the changes are in real time as the cells are growing. So multiple measurements can be done in the multiple times.
Okay, cool. Thank you. So this question is for both of you, actually, and so y'all can choose how you take it. So can you do the metabolite measurements of spheroids or organoids grown in Matrogen? So I guess
let me maybe start with this question. The short answer is yes. We don't show this data today, but we've done, and it's this work in progress. We've done some work on looking at how the signal behave if we put these samples in Metrogel. And there is some differences that we observed. For example, we tested real-time glow assay, and we saw some decrease in the signal. However, it is proportionate. So we still see a lot of substantial signal and, pretty high signal to noise ratio to be able to perform this assay in Metrogel.
Okay.
So yeah, I would add that we, haven't done any experiments here, at Promega, but there are several publications that have been published already, and that all those assays have been used with the Metrogel system.
Okay. Perfect. Awesome. Thank you. So this next one is for Katya. So this was from Estefania. She's asking, have you tried the Puma system Flowchip with native isolated islets? They are naturally 3D, but the cellular composition and diameters can vary. So can you use this platform to study this workflow?
So yes, we did tried it. We haven't evaluated the matrices yet, but we did some work on looking at secretions from the islets, and it will require certain optimizations because islets are very sensitive. But yes, we've done work with that.
Okay, cool. Thank you so much, Jolanta and Katya, for a lovely presentation today. And thank you all for attending, and please, have a good rest of the day. Thank you.
Thank you.
Thanks.